Okay, guys, are we ready? A little bit? Kind of. Okay. So, thank you guys for coming back. I would thought some people might not because I kept on threatening to make you leave. Again, if anyone wants to drop the class because you don't need it, drop it. It's not that I don't love you all, but I can let everyone on my wait list in except for maybe 2 people, and I don't want to not let everyone in. So, who wants to drop? No one? Come on. Okay. Anyways, so y'all love me. So, just a little bit of business before we get down to work. One, take the pre-test. Only about half of you have. You have until midnight tonight, well, 11:59 tonight to take it. There are 2 errors. So when I make the pre-test, I write the question, I write the answers, and then I have to click the right answer. And sometimes when I get disturbed by colleagues who want to chat or students who have important questions or my own brain seeing a bird fly by out the window or something, I don't click the right box. So there's actually 2, and someone emailed me about that. Thank you. Always email me when there's something wrong because I think I said something about the genetic code being something really not what it is. So if you guys are freaking out because you thought the genetic code was one thing and it was actually something else according to my test, don't freak out because you are probably right. I'm going to have to manually, unfortunately, go through and fix that. But always email me when something comes up that's so obviously wrong. And aside, someone emailed me thinking something was wrong with a question that was asking to translate a protein. So whoever that is, please come see me afterwards and we can talk through that. Okay, so that's that. First quiz attempt will be recorded. So I didn't say that, and some people took it multiple times and then emailed me when they realized they made a mistake or whatever. So I have it set that automatically the first time you take that quiz, that's the grade that gets saved. It says you have 3 attempts. That's just so that when the internet goes out or your roommate pukes on your computer or whatever happens, you can take it again. Well, if your roommate pukes on your computer, you have to go find another computer, and that might make things more complicated. But so don't take it 3 times. The first time is what's recorded. Email me when you do have trouble, and I'll say, go ahead, take it that second time. And I'll make sure to go in and double check that that second attempt is what I see for you. Questions about that? Okay. The weekly quiz begins this weekend, so don't just remember to get into the habit. Every time I post the quiz, you'll get an email that says the quiz has been posted. So hopefully that'll be enough of a reminder. But I definitely have had students in the past who constantly forget to take it. And that's a bummer because each quiz isn't worth very much, but altogether it's worth an exam grade. So if you forget them all the time, then that's really bad. And I'm going to be trying to have them ready by Thursday night, Friday morning. But that's not always going to be the case, but it will always be ready by 5 p.m. Friday. Again, recitation begins in 2 weeks, right? September 10th. Papers and discussion questions will all be on Blackboard. So beforehand, you are going to grab your paper and read it. Print out discussion questions. Don't bother answering them because that's what you're doing in recitation. But just read through that paper because you're going to feel kind of dumb when your group has and they can all talk about things and you have no idea what's going on. What else? Bring Scantrons recitation. I forgot to tell people this. It says on the syllabus that you need Scantrons for recitation quizzes and for exams. And you want to have them all by your first recitation because you're going to need to take a quiz at your first recitation. And if you don't have Scantron, you can't take the quiz and you don't get the points. See how that goes. So bring them, and I'm going to collect the exam ones and keep them, and I'll hand them out on exam days. Book. Right. So the book, for whatever reason, in the bookstore, it says that it's required. It's not required. You can learn cell biology from anything, right? You can go on Google and Google cell biology and learn it that way. And the other thing is Albert's, which I love, which is the book for this class, is not, it's available for free online. So why spend $100 and something? Now, some people don't like to read online. They like to have the book in front of them. So then you should buy it or rent it. Who rents their books? Right, that's cheaper. Do you guys go to Chegg or do you go to here? I think Chegg is really cheap, right? Who's compared? Anyone? Does anyone know Chegg? Yeah. Is it not any cheaper? Okay. Anyways, there was a thing on the radio about it. They didn't even do rentals when I was a student, and God, they kill you on those used books. But anyways, so don't buy it if you're strapped for money and you're okay with reading online. It's a searchable function on NCBI Books. The link is in the syllabus, and if you have any trouble with that, see me, and I'll show you exactly how to work it. Okay, I think those are all the things I forgot to tell you about. Okay, now I get to close the door. Okay, so this is where we ended. Reading the entire genome code. Listen to this, it's amazing. Who likes to read the whole genome? I had a boss who did. It was really weird. He did these sequencing projects and he would sit there with the sequences and just like reading them and finding magic in there. I had no idea how he did it and what he was doing. Socially, he was similar and like he just didn't relate to anyone. And big mustache, like soup strainer mustache. Oh, anyways, sorry, I was having a flashback. So the genetic code. What is the genetic code? Someone raise their hand and be brave. What is the, yes?
The series of nucleotides that translates to proteins.
Right, the series of nucleotides that's translated into proteins. And how many nucleotides does it take to make an amino acid or to code for an amino acid? 3. Okay, so genetic code, it's that translating the triplet codons into amino acid, right? That's real. That's where the real magic is, right? In the protein that's going to be made, and what that protein is going to go do. In my opinion, people who work on DNA and RNA and microRNAs might feel different. They might think the magic is there. Transcription, right? So you need transcription to actually get your RNA, so that you can then make your protein later, right? So that's your DNA being transcribed into RNA. So what's the purpose of transcription? Why do we do it? Just for fun? Is it regulated? Is it just willy-nilly, let's choose this gene? Anyone? Why would we, I see we're going to have a popular person in the front. Yes?
It's regulated.
Thank you. It's regulated. Who thought it wasn't regulated and just it was random transcription? Okay, good, because nothing's random. Well, a lot of things are random, but nothing's random when it comes to transcription. So it's highly, highly regulated. So who can give me an example of something that might turn on transcription of a certain gene? Just think about like what might make a gene be expressed. Yes. A former student, yes. Yes, when you eat a donut, because what happens when you eat a donut? Your blood sugar goes up, and that's going to cause a whole lot of things to happen, and all the genes involved in metabolism, glucose metabolism, and insulin production, and all of those things are all going to be turned on, right? Are those things going to turn on when you have low blood sugar? That would be dumb, right? Right, okay, so I want you to start thinking about things. Everything has a purpose. There's a reason for it. It's not just happening. These aren't these little finite little details that just happen in isolation. They actually all come together as part of this big system in response to something. So what's another example of something that might cause a gene to be transcribed, other than eating donuts? Yes?
When you transition into another stage
Right. So transitioning into another stage of the cell cycle, right? So if you're going into S phase, what kind of genes do you need to express? Okay, so the way to think, so this is giving you a little bit of a taste of the kind of questions you get on exams from me, right? So you need to stop and think to yourself, S phase, what is S phase? Synthesis, what happens during synthesis? DNA replication, what's needed for DNA replication? DNA polymerase. So there's one, right? So you might think there might be an upregulation and expression of DNA polymerase. What else do you need to copy your DNA other than just the enzyme? A primer, yeah, an RNA primer. So you might need to upregulate primase, right? So this is how you need to start thinking of like, I'm actually asking you to use information that you already know it. You guys all know this, right? Every time someone answers something, you're like, oh, I know that. So you need to start using it and putting things together. Okay, so I'll stop harping. Okay, no, this is its nucleus, not its cell phone. I don't know, I thought that was funny. Who thinks that cells each have their own cell phone? Do you think they'd have an iPhone or an Android? Android? I got one vote for Android down in front. Okay. I heard, on Facebook, I heard that Virgin Mobile has an iPhone with a plan for $30. Is that true? Anyone? No one. Because don't you all feel kind of dumb for paying a lot more than $30? Okay, I'm just saying. Okay. So the nucleus. So where is all of this happening? So we talked about DNA replication and DNA and gene expression and transcription. So where is this happening? In eukaryotic cells? The nucleus. That's a picture of the nucleus. Does it look like the nucleus to you? A little bit? It could easily look like a hairy cell, right? So this is the nucleus. So here we've got these little holes there. Those are nuclear pores, right? Because things need to go in and out of the nucleus. They don't just diffuse through, right? The nuclear envelope. It's a regulated process, which we will learn about when we learn about sending proteins to where they need to go to do what they need to do. And then the ER is right outside the nucleus. Now the ER is not this one small, tiny little thing. It's huge. But it is attached. It's actually attached to the nuclear envelope. And then you've got DNA and proteins. You've got chromatin. So all this is happening in the nucleus, right? Everyone feels good about that. Okay. In bacteria, it's not happening in the nucleus, but like the nucleoid. Okay, so you've got your RNA, and RNA could be many things, right? It could be mRNA, it could be microRNAs, it could be tRNA, it could be ribosomal RNAs, right? Okay, so we're going to focus just for a minute on the RNA that becomes becomes protein, that is translated to protein, which would be the mRNA, right? So that needs to get out of the nucleus, correct? Where does translation occur? Where? In the cell? Did I hear in the cell? Yes. Where else is translation? Where inside the cell? Someone raise your hand. I love hands in the air. It makes me feel good. Yes. Okay, so some not all proteins, but yes, you are correct in the ER because that's outside of the nucleus. Yes. Well, then so some proteins will be translated into the ER and then move into the Golgi, but very broad. Yes.
Ribosomes.
So are they translated in the ribosome or by the ribosome? By the ribosome. Where does the ribosome live? In the cytoplasm. Right. Generally, in a eukaryotic cell, translation occurs on ribosomes or by ribosomes, depending on how you think about it. In the cytoplasm, many proteins that will be going into certain pathways, like secreted out of the cell or into membranes or into lysosomes, are translated directly into the ER, right? Okay, so the RNA needs to go out of the cytoplasm. So that's what's happening here. right? Mature RNA is exported out of the cytoplasm where it's translated into protein. In prokaryotes, well, there's no nucleus, so it's a different story. So I talk about mature RNA. What does mature RNA mean? Does it mean that it's 21 and can get a beer? Does it mean that it can vote? What's mature RNA? I have it right here, right? Capping, splicing, polyadenylation, these are things that are required for maturation of RNA. So by capping, and this is very much a review, right? I'm trying to get you to the point of protein out of the cell, out of the nucleus so that we can start really learning. So capping, what's capping of RNA? What is that? Put a hat on it? Yes.
It's a signal tells it where to go to get translated.
Okay, kind of. Yep. So there's a 5' cap that's involved in export of the mRNA. Yep. And what's splicing? Yep.
Takes out the introns.
Yep. Removal of introns. What's polyadenylation? Yep. Adding on the poly A tail. Right. Okay. So does that happen in bacteria? No. Okay, good. Only in eukaryotic cells. So why is all this maturation? Why does all this have to happen? Why is capping and polyadenylation and splice? Why does it all happen? Yeah.
So it can exit the nucleus.
Right. So it can exit the nucleus. And why does splicing happen? What happens if you improperly splice your RNA? What happens to your protein? Yes? You'll have a different protein, right, because you'll have a different sequence of nucleotides in there. So what happens if capping, splicing, or polyadenylation don't happen correctly? Where will you find that RNA? Where in the cell? In the where? In the lysosome? Maybe. I don't know. I never thought about that. I never thought about how it's gotten rid of. I think we'll get there in a minute. But where? So if you think of the cell as two major parts, nucleus and cytoplasm. Yes? In the nucleus, right? Because you need the poly A tail and the 5' cap, and you need to be spliced properly to exit the nucleus. Actually, some viruses that have multiply spliced genes have ways of hiding introns so that they can get their gene out of, or their RNA out of the nucleus, because sometimes it needs to get out because the same transcript will make multiple proteins depending on how it's spliced. So if you were trying to isolate mRNA from the cell, what would you look for? Let's say, remember, there's a lot of mRNAs, right? A lot of RNAs. mRNA, tRNA, ribosomal RNA, microRNAs. What would you look for to tell you definitively this is an mRNA? Yes?
The tail and
Yep. Poly A tail and cap. So, and this actually, you use this all the time. So let's say you're trying to make cDNA. Who knows what cDNA is? Anyone want to take a guess? Yeah. Complementary DNA. So that's actually a copy of your transcriptome. So it's a copy of all of your RNAs in the cell. Okay? And so sometimes you make cDNA when you're looking at gene expression because it's a way to detect it. And the way you can isolate only mRNAs is you'll actually have primers to the poly A tail. And it'll only amplify polyadenylated RNAs. And so you'll only get cDNA that contains your poly A, your mRNAs. What if you're trying to isolate mRNA from a bacteria? What would you look for? Yeah. Anyone?
You probably look for it in the
Well, there's all cytoplasm, right, in bacteria, right? So pretty much you would probably think that most of the RNA you're going to find is mRNA in bacteria, right? Because you don't have, you don't have polyadenylations. You can't look at polyadenylation, so you'd have to go probably based on size and start and stop codons. Okay, are we having fun? Okay, so like I just said, mature mRNA is exported from the nucleus. And that's how it all happens. And if you want to learn about this in detail, you come take my 409 in the spring. But basically, here's your RNA, and you've got your 5' cap, and you've got your poly A tail, and there's all these proteins that bind to this, and they help to bring the RNA out of the nucleus into the cytosol, and then a bunch of translation initiation factors will then combine and help it bind to the ribosome. Okay, we kind of talked about that. So RNA message is decoded on ribosomes, right? Ribosomes. That's called translation. There's a lot of ribosomes that are present in the cytosol, and they are either free or associated with the ER. So don't think of it as just ER or just in the cytosol. It depends on the kind of protein that's being translated and where it's going, and we're going to learn that in great detail. So this is a cross section, right, of your ER, and see, each little dot here is a ribosome. That's a lot of ribosomes, right? So it's not like there's one ribosome and one mR. I mean, you want to think about how much is going on in the cell. Questions? Yes, no? Okay, so ribosomes are made of proteins and RNA. They translate the mRNA according to the genetic code. Here it is. So some people get confused because I tend to use the letter code, right? So here's your codons. Here's the amino acid that's encoded, and here's the letter code. So if you see a bunch of letters, look it up. They mean amino acids. So what's the effect of an insertion or a deletion in the DNA? Yes? It'll change your reading frame, right? Right. So it's really important not to have mutations and certain kinds of mutations and certain kinds of insertions and deletions and all those. It's pretty important to maintain stability of your DNA, I would say. Okay, so each codon has a tRNA, right? That's another kind of RNA, right, called transfer RNA. And the point of a tRNA is to match up to your codon down here and bind to your amino acid here, and it comes into your ribosome. It matches, the anticodon matches your mRNA, and the amino acid is transferred to the growing polypeptide chain. And then, of course, translation needs to start, so there are start codons. Everyone knows, right? Methionine, AUG, and stop codons that are at the end, UAA, UAG, UGA. I apologize. I tend to always say T instead of U, so just remember U is RNA. So how does the ribosome know which AUG to start at? There might be a lot of them, right? There's probably, when you consider there are only 4 bases in RNA, just randomly, there might be a ton of AUGs, right? So how does the ribosome know which AUG to start at? Has anyone ever thought about this? Who sat up late at night thinking, well, how does the ribosome know? I know someone has. I get stuck on all sorts of things. Every time I get on an airplane, I have a really hard time understanding lift. If anyone wants to talk to me about it, because that's a lot of metal in the sky. And I think about it while I'm on the airplane. And that's not good. Yes, do you want to explain lift?
It isn't,
Oh.
well, I mean, I sort of.
But do you really get it? Like, does it make you feel comfortable about flying?
Well, it's just because there's more air being forced underneath the wings, and it sort of creates like a.
Yeah, but like that's a heavy piece of metal.
Yeah, but it's a lot of like wing.
Yeah, the wingspan and all that. I know. Yeah, I guess. I don't know. I just don't feel good about it.
But isn't
And-
there like a promoter set or something before the first AUD?
So you don't want to use the word promoter because that's
Yeah,
transcription
that's
language.
all
But
right.
but I know what you're getting at. There's some sort of sequence before the first AUG. Does anyone remember what it's called? You can tell me bacteria or what? Did I hear Tatabox? No. Schindel-Garno in bacteria, right? And Kozak in eukaryotic cells. Yeah, so it just this kind of signals like, yo, here's the right AUG to start at, to the ribosome. Good, because it's important to start at the right AUG. What if you start at the wrong AUG and it's in a different frame, right? This is an important thing to think about when you are cloning. Who's done some cloning? I see one hand. Someone's done two. Okay. So you guys know when you're cloning, you need to figure out what gene's going into your vector. And if you have some sort of other AUG site or you don't have a Kozak sequence or something that confuses where the start site is, you're going to get the absolute wrong protein. The other thing is if you're making a fusion protein, sometimes when you clone a gene, you'll actually want a tag on it so you can detect it, and you want it to be in frame with your genes. So these are all really important things to think about. Okay. There you go. That's as much as you're going to see about translation in this class. Actually, I'm so bad because I totally ignore translation in this class, and I kind of ignored it in 409. That means translation's just not getting its due respect, so we'll have to fix that. I mean, we talked about it a little in 409, but not as much as we should have. Thoughts? Okay, polyribosome. So there's one message and many different ribosomes. So think about it. It's not just like a ribosome sits on the mRNA and goes down and makes a protein. Actually, it's tons and tons of ribosomes on one mRNA, and there's tons of protein being made. Something to appreciate. Okay, now what? So now we have protein, right? So what has to happen to this polypeptide chain? Does it just sit there? Who's thought about this? Come on, I know you guys are up late at night. That's why you signed up for 408, because you have all these questions that you're just begging to be answered. What happens to a polypeptide chain after it comes off the ribosome? Anyone? Yes?
Doesn't it get transported to
So some will go to the ER, absolutely. But what needs to happen to a string of amino acids so it can be a functional protein? Yes? It needs to fold, right? Because proteins, their whole function is based on their structure. They won't work and do the job they're supposed to do if they are not correctly folded, right? Does that make sense to people? Is anyone wondering what the hell I'm talking about? Who thinks about proteins as these little machines that go do things? Okay, someone is. Yep, okay, good. Some nods. Okay, so like for example, hemoglobin, what does it bind? Oxygen, right? If the structure, let's say there's some mistake and the folding of hemoglobin is all messed up, will it bind to oxygen? No. What does that do for your red blood cells? What does that do for the organism? It's bad, right? So just I want you to think about proteins as something that needs to fold and function properly and do something, right? Okay, so where does it go? So I heard ER, so some proteins go to the ER. What about those proteins that are involved in S phase that we talked about, proteins that are involved in DNA replication? Do they go to the ER? Why would we send them to the ER? Where do they need to go? Nucleus, right. What about a protein that's involved in electron transport? Where does that need to go? Mitochondria, right. What about some sort of acid hydrolase that's involved in degradation of lipids? Lysosome, right. Very good. Okay, so they go to their home, their cellular home. So every protein has a home where it does its job, right? And it's all part of making the cell work and do what it needs to do in order to make the organism or organ or whatever, however you need to think about it. Or like if you're thinking about a unicellular organism, it's a little bit different. But still, every protein has a job. It's just not a random protein sitting there. Okay, so what if there's a mistake in folding? What if there's a mutation? We kind of just talked about that, but what if there's a mistake? What's going to happen? What's going to happen to the protein other than it not functioning?
If it's a bad protein or a wrong protein, you want to degrade it and get rid of it.
So you would want to degrade it and get rid of it. So what do you think happens to a misfolded protein in the cell? So like just think about, okay, it's not folded properly. Remember, proteins have different characteristics, amino acids have different characteristics, right? Some hydrophobic, some, so normally when you have a protein that's folded properly, where would you find the hydrophobic regions? On the outside or on the inside of the folded protein? On the inside, right? It would be buried. It would not be exposed to the aqueous cytoplasm, right? Unless it's a membrane protein, and then it would be embedded in the membrane. So, but if a protein's misfolded, then there would be all these exposed hydrophobic regions, right? And then hydrophobic will bind to other hydrophobic, and then you get these aggregates of proteins that bind to each other and create a big mess, right? That's bad, and actually that is what is seen in a whole lot of neurodegenerative diseases. So protein aggregates are bad, and they tend to be highly ubiquitinated in the attempt to degrade them. Yep.
How does a protein actually get to where it's supposed to go?
Ah, you're going to learn about that in a way that you never thought you would. Oh, yeah, you will. You're excited, I know. Yeah. It's awesome, actually. It's very cool. It's very cool. Lots of vesicular transport and different signal sequences. It's going to be cool. The next few weeks, we're all over it. All of exam 1 is going to be that. Okay. So what if there's a mistake? Bad. Okay. So what's the point? What's the point of this all? Why am I telling you about this? Because it's my job. Okay.
Keep the cells moving.
Well, I'm trying to get you excited about learning about intracellular traffic and learning about proteins and where they go and why they need to go there and how they get there and what happens when it goes wrong. Because who cares if I tell you the signal sequence KDEL sends proteins to the ER if you're not thinking about it in the context of a larger picture of proteins having a function and a job and going to somewhere to do something, all in response to some outside stimulus and adaptation. Yes. Yes. What if there's a mistake? What if there's a mistake in folding? Tell me. What is your greater question? So if a protein doesn't fold properly, you'll have all these exposed hydrophobic regions. It just will not be right and will need to be degraded and frequently will be degraded by the proteasome. But sometimes when things don't work quite right, you'll have aggregation of misfolded proteins, and that is those are found frequently in neurodegenerative diseases. So that's an example. And actually, cystic fibrosis, the mutation in cystic fibrosis, transmembrane, whatever, it's a channel. That also, it results in misfolding, and I believe the protein can't get out of the ER. So it's not good. Mistakes are bad. Okay, any other questions? Okay, good. Here you go. Misfolded proteins are associated with disease. And so here's an example of Lewy bodies in Parkinson's disease. So you just kind of get these fibrils, and they form these plaques, and it's bad. But also misfolded proteins are associated with Alzheimer's, spongiform encephalopathies, type 2 diabetes, cystic fibrosis, and some cancers. So you want, it's not just problems with your DNA. It can be your problems just with protein folding, which sometimes result from insertions, deletions, or some sort of mutation. Yes?
If we know so much about the proteins and folding and the different things that help figure out which one's supposed to be which, why is it so hard to figure out the cure for it?
So if we know a lot about protein folding, why is it hard to cure these diseases?
Yeah.
Is that your question? We don't know that much about protein folding. Yeah, so that's part of the problem, and it's hard to fix things, right? So let's say you know that one mutation, like in cystic fibrosis, there's one major mutation that causes cystic fibrosis. So we know the mutation. Why can't we fix it? Well, how do you replace something that's broken? So you can put things, you talk about adding a gene back in, but the reality of gene therapy is actually really difficult, right? Because you need to get the gene to go to the right place and not cause cancer, because that's a problem. But yeah, so it's that's, I think, what's so frustrating about research, which is we know so much, yet we can't, it's hard to get from the bench to the bedside, basically. Yeah. Okay, so how are we doing on time? Very good. Okay, dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet. So we're going to talk for a minute about cell cycle and mitosis. And so we talked the other day a little bit about chromosome condensation, and that's, of course, in the first part of mitosis, right? Because you need to condense your chromatin to become chromosomes. Because if you don't, what happens? You'll break your DNA when you go to separate, right? Isn't it so much easier to separate 2 bananas that are next to each other than to separate a bunch of tangled spaghetti? Yeah. So I just equated chromosomes to bananas. Great. Okay, so let's talk about the cell cycle. It's important, right? No. Okay, so the cell cycle has the 4 main phases, but I would like to say it's basically mitosis and other. So there's mitosis, G1, S, G2. There's a fixed amount of time for each event to be completed. If things take too long, that usually means there's a problem, and there's whole cascade of signaling happens to kind of stop things and repair things, and you need to go in the right order, right? You need to do S phase and then M phase, right? Right? Because if you don't replicate your DNA, what will you put in your other cell when you go to divide, right? Each event must have a start and an end, right? You need to finish S phase before you can go on, right? The cycle has to have backup to ensure there's control in case of system failure. So if you've got one protein that's involved in regulation of S phase, just one protein, and something goes wrong, that's horrible, right? So there's usually a lot of different measures of control. And then it has to adapt to different cell conditions and different cell types, right? So is the cell cycle the same in skin cell, or does it occur with the same frequency, I should say, in a skin cell or a neuron? No. In a kidney cell or a liver cell? I mean, it's different cells are going to have very different timetables for their cell cycle. And some don't divide at all, right? So just things to think about. So there are these checkpoints, right? So checkpoints are points in the cell cycle where the cell stops and says, okay, is my DNA replicated, right? Is everything favorable for me to enter mitosis, right? Because if my DNA is not replicated, should I be entering mitosis? No. There's a checkpoint before you exit M phase, right? Because you need to make sure your chromosomes are attached to your spindles, right? Because the spindles are going to separate your chromosomes. And if they're not attached and you move forward, what's going to happen? You're going to have unequal numbers of chromosomes in each cell. And when you start to have the wrong number of chromosomes in each cell, what happens? Is that good? No. Frequently, that's associated with cancer. Frequently. And then, do you want to enter S phase if you have lots of DNA damage? No. Do you want to enter S phase if you're in starvation or some sort of very bad environment? No. Okay. So we're going to talk about the cell cycle and cell cycle control in Unit 4 when we talk about controlling cell numbers. We're going to talk about the things that regulate the cell cycle because, again, everything is regulated. If there's anything that you learn from this class, it's that everything is regulated. Nothing happens for no reason. Everything occurs in response to something. What happens if the cell cycle is not regulated? Cancer. I'm hearing lots of cancer mumbles. Yeah, cancer. Unregulated cell cycles. Bad. Right? You get things are being broken and genome instability. It's bad. What mechanisms exist to protect an organism from cells with a dysregulated cell cycle? There's a major mechanism that protects the organism from cells gone bad. Yes? Apoptosis, right. Cell death, right? So if the cell cycle becomes dysregulated, usually it will start, P53 is a big one. It'll activate a whole lot of things, though, that will turn on apoptosis, and the cell will commit suicide to save the organism. Isn't that nice of a cell? Yep. So how does the cell end one phase of the cell cycle and start another? Does anyone know anything about that? We're going to learn a lot about that. It's all regulated by ubiquitination. Well, I'd say about very much regulated by ubiquitination and degradation of certain proteins. And so I love ubiquitin and degradation of proteins by the proteasome. So anytime I can throw that in there, you're going to have to learn it. So we're going to learn about that at the end of the semester. Here's where you screwed up. Telophase comes after metaphase. So since we're talking a little bit about cell cycle, I figure we'll talk a little bit about mitosis in the context of cytoskeleton, because we're going to spend a few weeks talking about the cytoskeleton, right? So we talked about the cytoskeleton structure and this and that. A big thing, though, is microtubules form the mitotic spindle. So here's your blue nucleus and your microtubules. And you can see as you go through, right, so your chromosomes start to condense and they line up. And look at what happens. This kind of disorganized looking, it's actually highly organized, but disorganized looking microtubules, they actually reorganize and become mitotic spindles, and they attach to the chromosomes and they spread them apart. And so there's a huge amount of regulation there in terms of microtubule dynamics. We'll learn all about that. So what role does the cytoskeleton play in mitosis? We just talked a little bit about the mitotic spindle. So how could understanding the role of the cytoskeleton in mitosis lead to cancer drug development? So let's think. We're talking about microtubules and formation of the mitotic spindle. How is this relevant to you're working in a pharmaceutical company and you're like, I gotta cure cancer, right? Isn't that the goal? if you're in a pharmaceutical company, if F science, right, cure cancer. So how could understanding the cytoskeleton be useful in drug design and curing cancer? Has anyone ever thought about this one? I know someone has. Yeah.
Maybe if you understand more how the cytoskeleton works. In the case of cancer, maybe you could have also maybe drugs that will target cancer cells and
Okay, so drugs that specifically target cancer cells. Let's get more specific, right? Let's talk about the mitotic spindle. Yes.
Well, if a cancer cell is unchecked growth and reproduction, the cytoskeleton assists in the division. If you don't have a cytoskeleton, you can't divide.
Right. Okay, so if a cancer cell is unchecked in growth and reproduction and cell division, and you need the cytoskeleton for cell division, you don't have a cytoskeleton or not a functional cytoskeleton, which is where I'm trying to push you guys, then you can't divide. Now, if you could somehow mess with those microtubules so that they couldn't separate those chromosomes, that would be pretty cool, wouldn't it? Has anyone ever heard of Taxol? Has anyone ever known anyone who's had breast cancer? Yeah, isn't that so? God, I hate that. That question. It's so many hands that go up. Yeah, it's but Taxol. They were probably treated with Taxol, and that's probably Taxol and Herceptin. I think are the two big treatments for breast cancer, and Taxol actually binds to microtubules and inhibits their disassembly, which inhibits their function. And so you can't go through mitosis. And so remember I said there are all these checkpoints if the cell cycle isn't working right, and that kind of turns on a whole lot of alarms get set off, one of them being like, hey, something's wrong, apoptosis. So what happens is you treat someone with breast cancer with Taxol. It inhibits the mitotic spindle from functioning. It inhibits cell division, but it stalls it at a weird spot. I think it also inhibits attachment of the mitotic spindle to chromosomes. And then the cell goes, holy cow, not good. Apoptosis. And that's great, right? We want apoptosis when we're talking about cancer cells. Right? Right? Yep.
So if you're treating somebody with a drug like that, what keeps it from basically wreaking havoc with the entire rest of your body that if you get a cut, now your cells at the cut need to
Great
survive?
question from down in front. Yeah, so what keeps this drug from basically killing your whole body or being highly toxic? Well, it is highly toxic, and that's why people on these drugs are really sick and immunocompromised. So what it does is it attacks the cells that are dividing the most, right? So it's attacking cancer cells, it's attacking immune cells. So that's why you become immunocompromised. You get skin issues. I had a friend who was on something and her skin was a serious mess because your skin cells are dividing. I mean, so it's not good, right? But the good thing is, like, your brain's going to be in better shape. I mean, so yeah, so chemotherapy, it's not ideal. That's why a lot of these antibodies, so Herceptin is an antibody against a specific receptor that's overexpressed in cancer cells. That's nice. Targeted, right? So it's better. But yeah, so you're absolutely right. So I guess I should let you guys go away. Okay, so anyone on the wait list, please come up here.